Students, this is Pallavi Mohan, and today we'll be discussing another science topic, mutation. We have requested Dr. Aruna Mohan, retired professor, Delhi University, to take up this topic and discuss it with you. Over to her for discussion.
Thank you, Pallavi. Dear students, mutation is an interesting topic. It is sudden change and spontaneous change in genetic material. We can put them in two categories: point mutation and frame shift mutation. Point mutation, where there is mutation of one gene or one amino acid. Either one gene is mutated or one amino acid is replaced by another amino acid. These are point mutation, and all the Mendelian disorders come in this category. Some of them we have discussed in previous episode. The frame shift mutation, what happens? Either full chromosome is added or deleted, or big piece of chromosome is added or deleted. You know, students, that chromosome has two chromatids, and each chromatid is having full length DNA end to end, and genes are located on this DNA. So if there is big chunk or big piece, it comes from this DNA. That means it will be added to one chromosome, then other chromosome will be shorter than the first one. Or one full chromosome goes to other haploid. We discussed Down syndrome, 21 trisomy, where we had one extra, 21 number chromosome. Similarly, there is Edwards syndrome, that is chromosome 18 trisomy, and then there is a Pallavi syndrome, 13 number chromosome trisomy. Now, Down syndrome we have discussed in previous episode. I will request Pallavi to explain Edward and Pallavi syndrome.
Sure, Dr. Mohan. So far we are discussing mutation. The Mendelian disorders are sickle cell anemia, thalassemia, cystic fibrosis, phenylketonuria, hemophilia, muscular dystrophy, color blindness, and so on. The chromosomal mutation, now this is important to note. Let me just adjust the frame. Okay, so there are 5 types. One is insertion, duplication, deletion, inversion, translation. Now in inversion, if you see, this is the original one, A, B, C, D, E, F. Now in inversion, you see A, B, and then you see the inversion. So it is E, D, C, F, which is inversion category. The second is duplication, in which original is A, B, C, D, E, F. However, the duplication occurs right at D. So it is A, B, C, D, D. D, D is the duplication, E, F. The third is deletion. So the original one again is A, B, C, D, E, F, and deletion is A, B. We can't see the C. C is the one which is deleted. So A, B, D, E, F. C is deleted. So it is deletion. Insertion. A, B, C, D, E, F is the original one. A, B, C, D. What is here? It's L. So L is the insertion. E, F is the original. So that is insertion category. Then comes translocation. Now here there are 2 strands which are there. A, B, C, D, E, F and I, M, N, O, P, Q. In this translocation, you see A, B, C, and then it should have been D, E, F. Now, since it's a translocation, this one went here and this one went here. So D, E, F is below and O, P, Q is right here in the category. So this is chromosomal mutation. We now move to Edwards syndrome and Pallavi syndrome, which is trisomy 18 and 13 respectively. Now, if we see the signs and symptoms, so basically we are talking about the trisomy. So let's go straight away to the signs and symptoms, which is clenched fist with overriding fingers. You can see this picture which clearly shows that, and it shows the trisomy 18. Small fingers, short sternum, club feet, heart defects, and kidney defects are other signs and symptoms for the same. Pallavi syndrome is basically trisomy 13. Let's go ahead and see that as well. The symptoms and features. Now, all these are the symptoms: abnormally small eyes, absence or one or both eyes, reduced distance between the eyes, problems with the development of the nasal passage, extra fingers or toe. This is one which we will discuss right now, and I'll be showing you pictures for it. Here comes the next slide. Patau syndrome or trisomy 13. The particular one is, if we count here the feet, the toes, it's 1, 2, 3, 4, 5, 6. In this category also it is 1, 2, 3, 4, 5, 6. So as you see, it should have originally been 5 plus 5, but here you see 6 plus 6, which is 12. So now that is an example of trisomy 13. Going back to Dr. Aruna Mohan for further discussion on Huntington's disease.
Students, Huntington's disease is a special feature of this particular episode. In this case, there are repeated sequences of triplets. When sequence is over, after that it is repeated 35 times, 40 times or more. With the result, it results into disorder, and it is in the brain where it is degenerated, that area, or depressed or not grown properly, and it happens in the basal ganglion, and it has many other features like symptoms which Pallavi will explain. This is Huntington's disease.
Sure. So thank you so much, ma'am. As ma'am explained, this is the normal brain section, and this is the Huntington's disease, in which you see enlargement of the frontal horns of the lateral ventricles. Now we move on to the next one, which is the symptoms. So these are the symptoms, which is difficulty in concentrating, memory lapses, depression, that means including low mood or a lack of interest in things, and feeling of hopelessness, stumbling, another mood swings, and irritability. So these are the symptoms which are particularly to this disease. We request you, students, that in next episode we are coming up with free classes for class 11th and 12th class students. So we request you to please subscribe and also hit on the bell icon, so that you automatically know whenever we are putting new classes, and they are all free for you. It is basically class 11th and 12th. Thank you so much. Bye for now.